Hello friends, in this video we will be discussing GAT-B 61st to 100 questions. So 61st question is a cDNA encoding human protein of interest was cloned in bacterial expression vector and introduced into bacterial cell for expression, but no expression of human protein of interest was obtained. This was because. Okay, so let's do this. See, what is done is, suppose this is a human DNA and from this human DNA RNA was made. Okay, now from this RNA they used reverse transcriptase and produced cDNA. Okay, now they've introduced this cDNA into the bacteria. Okay, using the expression vector. Okay, now it was found this cDNA was not able to produce protein in the bacteria. Why? So what happens is, as we know, there is a codon. The codon is universal, right? But in the prokaryotes, they prefer certain codon rather than using another codon. Suppose I'm just giving an example. GAG, AG. Okay, these both are coding for tyrosine. Okay, so what happens is, in species to species, everywhere it is producing tyrosine, GAG and CAG, but bacteria may prefer GAG than preferring CAG. It would not prefer CAG to produce tyrosine. Rather, it would just prefer GAG to produce tyrosine. Okay, so this phenomena is called as codon bias. Okay, so here third option is the correct option. Okay, coming to the 62nd question. The protein code covering the nucleic acid in virus is called as. Okay, so we see here there is a virus. Okay, a virus here, this coat, as we can, this coat is made up of protein, and this is called as capsid, and it is made up of capsomere, and it is a protein coat. Okay, and if this protein coat is covered by an envelope, this is called as enveloped virus. Enveloped virus. If this envelope is not present, it is called as naked virus. Okay, so protein coat covering the nucleic acid in virus is called as capsid. So this protein coat which is present here is called as capsid. So fourth option is the correct one. There is a mutation in the Operon repressor in E. coli in which prevent its binding to the operator. What is likely to happen when level of glucose is low in the cell? See, low glucose in the cell, first point that we have to note, if there is a less glucose, the bacterial cell is going to produce a glucose. Okay, so now there is a mutation in the Operon repressor. Okay, let's do this. Okay, so I'll be just doing it here. Suppose this is a Operon. Okay, there is no glucose, so we want glucose. Here is the operator, and there is a, before this is a, okay, just let me erase this. First there is the presence of, okay, so there is a promoter and then there is a operator, and there are here X, Y and A, sorry, Z, Y and A. Okay, now here On the operator, there is a bound repressor. Okay, so what will happen here? RNA polymerase cannot can bind, but it cannot transcribe these genes, right? But they are saying that repressor is having mutation. That means it will not bind to the operator now. If it is not binding to the operator, there is the transcription of these genes, right? So constitutive expression of these genes happen. Okay, understood? The repressor, because there is a mutation in repressor, this repressor is removed from here, and this RNA polymerase is going to transcribe these genes, and now glucose is produced. Okay, now let's see this. So there is a constitutive expression of the lac operon. Next question. Which among the following is character-based method for construction of phylogenetic? So this is a conceptual question. The character-based method for construction of phylogenetic is the maximum parsimony. Okay. Next is a denaturation of protein leads to the loss of biological activity mainly due to. So whenever because of heating or any chemical agent, what can happen? The protein structure may denature, and denature causes the loss of function of the protein because it has lost its secondary and tertiary structure. So first option is the correct option here. Next, which of the following disease is transmitted by a mosquito bite? So this is again a conceptual question. Malaria, dengue, yellow fever and filariasis. Filariasis is also called as Wucheria disease. Okay, these all are caused by a mosquito bite. Next question. Which of the class of enzymes do not participate in the glycolytic pathway? Isomerases, yes, this participates. So this is wrong. Liases also participate. Transferases also participate. Ligases, no. Ligases do not take participation in the glycolytic pathway. Ligases do take part in replication of DNA. Okay, so this is the correct option here. Next is Rushton disk turbine is widely used in stirred tank fermenters to mix the culture broths. The type of flow pattern generated is radial flow. Okay, so this is a conceptual question. Okay. Next is plant gum is composed of different polysaccharides except. So plant gum contain galactomannan, xylem and galactan also it contains, but starch is a food. Okay, it is not there in the gum, so starch is a correct option. Next, horizontal gene transfer in bacteria can occur through. Okay, so horizontal gene transfer can occur through conjugation, transduction and transformation. See, what happens in the case of conjugation? Here is a bacteria. Here it forms a sex pili and conjugates with another bacteria. This is one first bacteria, this is second bacteria. It can transfer its genome here. So this is conjugation. Okay. Second is transduction. This is the bacteria and here the virus can sit and transfer its genome horizontally into the bacteria. So this is called as transduction. Okay. Next is transformation. Transformation is if a bacteria is present, okay, and now in its surrounding any genome is present, and now if this genome is compatible to this bacteria, it can take up this genome within this cell. Okay, so this process is called as transformation. Okay, transformation we see in a Griffith's transformation. Griffith's transformation principle was given. What was the bacteria doing? It was taking up the genome from its surrounding. Okay. Yes, TCA cycle operates under aerobic conditions. Reactions catalyzed by which of the following enzyme requires participation of molecular oxygen? Succinate dehydrogenase, isocitrate dehydrogen, none of the TCA enzyme requires participation of molecular oxygen. Yes, this is correct. TCA cycle, none of the enzymes require the molecular oxygen for its function. So, none of the TCA enzyme require participation of the molecular oxygen. Next question, which of the following is not an example of RNA virus? So, here we see rabies virus is a RNA virus. Okay, vesicular stomatitis virus. This is also an RNA virus. Ebola virus is an RNA virus. Whereas SV40 is a DNA virus. Okay, so this is the correct option. Next, tDNA carries which of the two phytohormone biosynthetic genes? See, what is tDNA? tDNA is also called as tumor DNA. Now, what is tumor DNA? Tumor DNA is going to it is also known as viral DNA. It is this part of the DNA is containing all the information to infect the host. Okay. So now tDNA contains auxin and cytokinin. Okay. What does they do? Whenever they enter the host, they can cause infection. Okay, but they also need their growth factors. Okay, they also need food and everything, so they produce food and everything using auxin and cytokinin. These are the growth factors for tDNA. Okay, so tDNA is there. It may contain auxin gene and cytokinin. Now they will produce this. Okay, and growth, it will help in growth of uh, the whatever the viral gene it is. Okay, which one of the following amino acids give yellow color on reaction with ninhydrin? So it is a conceptual question. Proline gives the yellow color when reacted with ninhydrin. Next question, what is the number of hydrogen bonds in a double helical bDNA structure